Okay, good, good afternoon. Can you hear me all right? Okay. We'll begin, and I want to thank those of you who have braved the elements, particularly our guest, Sangeeta Bhatia, who came from up north where they really have snow, not just this trivial stuff that we've been fighting with. But we'll get started so that we can perhaps finish a little earlier if the weather is more inclement. So you all know, I mean, today's discussion is about malaria. Really one of the, I guess you'd call it, great diseases of mankind. The organism is said to have been around, the parasite, for perhaps 100,000 years. But the disease, malaria, bad air in Italian, really took off when society changed and you began to have people living in towns and together rather than nomadically wandering around, and was greatly influenced by changes in human sociology and economics. It was also greatly influenced by some of the catastrophes of mankind, namely wars, a disease of predominantly the equatorial countries of the world, but it wasn't always that way. And I found this poster from the 1940s, I guess, about the United States. In fact, if you read about the history of Washington, D.C., one of the reasons the capital was chosen as that site, aside from trying to make Mr. Jefferson and the Virginians sort of content dealing with the industrial Northerners, one of the reasons was that Washington was a swamp. And in the course of building the city, malarial outbreaks and epidemics were very common. The key, of course, is the Anopheles mosquito, and the areas in red are the areas in the United States where, I think this is in the late 1930s or 40s or something, where it was very prominent. But there were cases that were present in other areas, because wherever the mosquito could flourish in water, fresh water, the disease was relevant. The early treatment of quinine coming from the bark of the cinchona tree was for many years the only thing, and at the time of this poster, as it says, it was the worldwide remedy. And for the correct dosage, see your doctor or health officer. Now, I found several interesting posters which date to the 1940s. This is the Second World War. And one thing was an awareness that malaria was a major killer, not only of people living in the equatorial African areas as natives and so forth, but soldiers from any country who went into those areas and didn't take any precautions, it was very deadly. It's estimated that 500,000 American soldiers during the Pacific Campaign in the Second World War became infected with malaria. And of them, somewhere between 50 and 60,000 died. This is a lethal disease. So here's one poster, the peril. The mosquito is more deadly than the enemy. And don't go to bed with a malarial mosquito. Sleep under the covers and under the net, some of which hasn't changed too much, which we'll hear about a little bit later. But the was that if you covered yourself up, because these mosquitoes came out at night, and it was particularly the female mosquitoes, you remember from dengue, that ingest blood and transmit it. The males are vegetarians and live on nectars. And you shouldn't spoil a good dream by failing to sleep without a bed net. So this was the level of sort of popular education that was given to the military. The Japanese were, well, this was another one, don't be a dummy, avoid malaria. But the Japanese were more dramatic in their posters, recognizing the linkage to death. The American posters, although they delivered a message, were a little bit more like a comic strip. So amongst the big problems that come to mind, which will be addressed today, and are sort of things that rode through my mind, is sort of, why can't we vaccinate against this disease? And how do you go about developing a drug when there's a very complicated life cycle? By the way, malaria is the only disease for which 4 Nobel Prizes have been given, and there's a 5th one out there, I think, for whoever develops the effective vaccine. And the history of the Nobel Prizes is something you might enjoy reading, because it really is the history of modern parasitology, and particularly malaria. One of the Nobel laureates is not known for his work on malaria. That's Golgi. He shared the Nobel Prize with Cajal for the anatomy of the nervous system. But his studies of malaria pointed quite clearly to a vector in a mosquito. So one of the things the parasite does is live in the liver for quite a while. And how does that work? Can you replicate this in culture? It's not so easy to culture mammalian hepatocytes. We'll hear more about this. And what are the cellular and immunologic mechanisms for the different phenotypes? Not everybody dies. People do become resistant. You do get reinfected. How does that work? And most importantly is, what are the global public health initiatives that are going on directed against this major human killer? Not so much in Washington. We have other dangerous things in Washington, but much more in Africa and in parts of South America and so forth. So that's what our 2 speakers today are going to address themselves to, and other things. So I'll briefly introduce them to you. Our first speaker is Tom Willems, who took his MD and PhD degree at the University of Chicago and a residency at the University of Pennsylvania. He came to NIAID in 1984, has done an enormous amount of work on mechanisms of malarial infection, the development of drug resistance, and this is a huge problem, which he will speak about. And it was largely for this work that he was elected to the National Academy of Sciences and the Institute of Medicine. He's the director of the Malarial Genetics section in NIAID. And our guest today, Sangeeta Bhatia, took her medical training at Harvard and her PhD at MIT. Sangeeta is a bioengineer. From my reading, she's one of the most outstanding people in American medicine that's trying to link engineering principles to major biological and medical problems. And her primary interest has been to develop technologies that would lend themselves to widespread use for a whole range of things, which she will discuss with you. But in particular, she'll tell us about some of the exciting work that she and her colleagues at MIT have been doing dealing with malaria. It's noteworthy that amongst her many, many, many accomplishments, she's been widely recognized for her role as a mentor, her interest in education, in science, of young people, even in elementary school. And most recently, last year, I think it was last year, she received the Lemelson MIT Prize, which was given for dedication to the next generation of scientists. So we're very delighted that Sangeeta braved the elements. I almost hope it snows like crazy so you'll stay here for a few days with us. At any rate, enough of me. Tom, would you please?
Can you hear me well back there? Yeah. Wynn, thank you for the introduction. And to those of you in the audience, thank you for coming through the storm and the coming precipitation and weather to just be so supportive and listening to our talks this afternoon. Today I'll talk about not only malaria interventions for global health, but I'd like to focus, and it's be a large component of the middle third of my talk, on the importance of long-term scientific discovery, fundamental research to the progress we're making against malaria. A good point of departure is the Millennium Development Goals. These were announced in September 2000 by the United Nations, made the cover of Time magazine at the time, and addressing across the globe the importance of eradicating extreme poverty and hunger and achieving education, advances in education, gender equality, empowerment of women, reducing child mortality, improving maternal health, combating HIV/AIDS, tuberculosis, malaria, ensuring environmental sustainability, and developing global partnerships for development. Malaria is at the center of a lot of this. Now, as we look at this, to back up just quickly on that, is to think that one of the goals, how do I reverse? Yeah, we're trying that. Here we are. Not the volume. The screen? I'm missing it. Yes. Yeah. Oh, we're at the end. I want to reverse it. If you hit the components we use for, it's always going to go forward. Yeah, there we are. Okay. Good. So you have to hit the arrow button. I'll just use that. Thank you. Right. So down at the bottom here, emphasis of this article was the reduction by 2/3 of the mortality rate in children under 5 by the age of 2015. How have we been doing? Here is a curve published in The Lancet in 2014, period of 1970 to 2013. And you can see a reduction in this period of from about 140 deaths per 1,000 live births down to less than 50. A reduction of almost 2/3. We want to, the goals are to move even further. The goal in 2000 was 2/3. We've achieved about half. And no, this is for all deaths. We'll come to the case for malaria in a moment. Also, as part of these studies, came information on the contributions to the change in under 5 deaths, particularly in the period since 1990. If you look at these, you'll see a contribution for maternal education of nearly minus 40%. Of income, advances in income, socioeconomic status of about minus 16%. Reduction in the increase in fertility, actually increased population growth, has given a figure of plus 25%. And then there's this secular trend, you see, of minus 72%. Now, when I was looking at these studies, I scratched my head. What do they mean by secular trend? Well, I put a footnote down here. A big factor in secular trend is the development of new technologies, along with improvements in healthcare systems and public health policies and health initiatives. New technologies include vaccines, drugs, and diagnostics, and in the case of malaria, intervention, insect-treated nets. Minus 35 in education, minus 38.5 in education, and minus 15 in, with improvements in income is at almost minus 50%. Just pause a minute and think about that. Just contributions of education and improvements in income and what they can do for improvements of global health. Now, let's look at this 1 to 4 year old category of children. Figures from 2010 shown in this slide. You see a number of contributors to these causes of death. Down in the lower left, malaria contributes almost 21%. Now, pause again for a moment and think 21% contribution to malaria in this 2010 period. Has malaria reduction in deaths with malaria tracked with what I showed earlier in that period of impressive decline since 1970? Here are some figures. You can see here that since 1990, the incidence and the deaths have risen slightly, period of 2000, 2005, and then come down again. In recent malaria reports, World Malaria Reports from UNICEF and World Health Organization, there's much cheering about the reduction and the achievements of control against malaria. And some 30 to 50% in some cases. But if we look at the longer picture from 1990 and forward, there was increases. Now we know many of them were due to drug resistance, and then these decreases with recent implementations of controls that bring us back to the figure about 1990. Let's look at the longer term. Since 1900, about a century's worth of data here. This work, again, from World Health Organization, Richard Cartier and Kamini Mendis in an article published in 2002. I've split out the data in that paper into two areas. The area of sub-Saharan Africa, which is shown in the red, and then the area outside sub-Saharan Africa, which is shown with the black bars. You can see from the black bars that there has been a dramatic reduction in deaths, annual deaths per 10,000 in this slide, since 1900 in areas outside sub-Saharan Africa, particularly in the Pacific Rim, areas of China, South America as well. Do you know that 100 years ago, in areas of Canton province in China, the death rates and the incidence of malaria approaches what we see in malarious regions of Africa today. And much of that has been eliminated. And I'll come at the end of my talk as to some of the reasons why that elimination has occurred. Sub-Saharan Africa is a different story. There was a reduction of, it always has been a slightly higher death rate than outside sub-Saharan Africa. But beginning in World War II, there was some drop in malaria. And then in the 1970s, when it really took hold, a reduction down to about 50% of what it was in sub-Saharan Africa. Then a return by late 1990s and that 2000 peak I showed earlier that approached rates that were present, were occurring at the period of World War II. So this longer-term perspective gives you an idea of what's been going on in the past century in malaria control, with a dramatic reduction post-World War II, a rise, and today a reduction again that we hope can continue and bring it down. We have yet to achieve this reduction that was had been accomplished in the 1970s. Much of this due to the drug chloroquine. Here is a map, a regional map of the malaria endemicity between in 1900 compared to 2007, published in Nature in 2010. You can see here in map form what I was showing in the bar graphs earlier, that high area of endemicity, particularly in the Pacific Rim, southern China, extending out into Oceania, and in South America. By 2007, much of this is reduced in South America. Much of it is gone from China. In fact, in those highly malarious regions in China 100 years ago, you can hardly find a case of falciparum malaria today. Also of interest is what Wynn alluded to at the very beginning of his introduction, the cases in the United States. And you can see here they were predominantly in Southeast United States, although they were out in New Mexico and Oregon as well, Midwest. And today, you know, it's only malaria that comes in, imported malaria, we call it. So let's back up 100 years ago. That was a period of dramatic discovery in malaria research. It had just been shown that Anopheles mosquitoes transmit malaria. And discoveries of Ronald Ross and Battista Grassi in the 1890s, proven by this trio of fellows, an international group immortalized in this famous cartoon. These decided to do a study. What would happen if, you know, many people in many generals in the armed forces doubted that mosquitoes were transmitting malaria. Oh, no, that couldn't be the case, little bug. And these fellows raised the question, well, if we go and live in a highly malarious area where everybody's catching malaria and live in a hut for 3 months, shielded by screens, don't allow any mosquitoes bite or we're going to come down with it. So they marched in. They took a bottle of quinine, shown here in the center, just in case. And after 3 months living in the Roman Campagna, highly endemic for malaria, they came out without a case of malaria. Case proven. That area of Italy had been rife with malaria. You can read in Scientific American and other journals of the case in the Roman Empire of epidemics that may have contributed to the decline of the Roman Empire in parts of Italy. In area near Rome, there is this well-known stonework outside a church. It appeared on the cover of Clinical Infectious Disease in 2012, calling for alms for the poor who were struck down in the Campagna by malaria in an epidemic in 1694. With the discoveries of the anopheline transmission of malaria by the anopheline mosquitoes, also the availability of the quinine remedy at much reduced prices and in larger amounts, Angelo Celli began a campaign to reduce malaria among the workers in the Campagna in the early 1900s. He, I think, is one of the great heroes of malaria, an uncelebrated hero of malaria research and global health. He worked by the motto that to take care of one thing, but don't admit the others, and that's a translation of the Italian here. He saw that more than just medicine and mosquito control was the importance of caring for the health of the worker, their housing, their education, and their socioeconomic development. And as a result of this, malaria was much reduced, and the impact and the death rates were much reduced in these populations in Italy. Also, following these fundamental discoveries was a success in the Panama Canal, led by William Gorgas. Here's a picture of the Ancon steamer passing the first ship to go through the Culebra Cut of the Panama Canal. This was in August 1914. It would have made the headlines of the New York Times, but it was on the second page because of the outbreak of World War I. How had he done this? The French had tried to put the canal through in the 1870s under de Lesseps, the same fellow who led the Suez Canal successfully, but they failed miserably in Panama Canal because of malaria and yellow fever. One of my ancestors perished in that effort in the 1870s. Mosquito control and a quinine distribution program, up to 40,000 doses per day, made this success possible. And you can see the reduction in malaria incidence was down from 800 per thousand in 1906 down to 16 per thousand in 1916, concomitant with the control of yellow fever. Now, quinine was a backbone of many of these programs. It was developed from the cinchona bark that had been discovered and brought in from Peru in the early 1600s into Europe. And if you want a fascinating history in pharmacology and some chapters that make great reading, read about the history of quinine and how it came through in Southern Europe and in Northern Europe. And it was tied in with religious conflicts and doubts about the value of medicine and what have you, and even differential diagnosis came into play, the development of differential diagnosis in internal medicine. It wasn't a great drug, though. It was expensive, as I've pointed out. It also had side effects. If any of you are taking quinine, the effects of synconism, the ringing in the ears, and the effects on the GI tract are really unpleasant. And use prophylaxis against malaria is very difficult to achieve. So a new drug was needed. It was the first glimmers that it might be possible were through the German organic dye industry and the organic chemistry that developed in the late 1800s. Paul Ehrlich found that methylene blue was effective. That was announced in 1891. And through synthetic efforts, through the period of World War I and then into World War II, came other drugs with some of these similar aromatic ring structures that were, you can see the similarity to quinine. Pamoquin in 1926, Meproquid in 1931. These drugs were also unpleasant and had side effects, including yellowing of the eyes and then the GI disturbances, and also among troops, particularly in the South Pacific Campaign in World War II, Meproquin was rumored and widely, and as happens even in today's society, repetition of the rumor lends credence. We see that in the vaccine opposition groups, et cetera, today. And Meproquin was rumored to have even more disastrous side effects than just GI disturbances and turning the skin yellow. None of them true. Nevertheless, the continued development of this led to the discovery of chloroquine, initially in 1936 and its rediscovery during World War II. This was a dramatic drug, inexpensive, easy to administer, easy to manufacture, readily available, readily stored. And its global health impact by the 1970s, as I alluded to earlier, was tremendous. I think it's arguably, if you look at the handful of drugs in the 20th century, medicines that had the biggest impact on global health, chloroquine makes that list of the top few and may be the leader. And I'm including in this discovery of the sulfas and penicillin, the anti-tuberculosis drugs. Its impact was enormous. Now, Also in this period, around World War II, 1930s and into World War II, was the discovery of another very dramatic compound. It had been known since the discovery of Anopheles mosquitoes, of course, that controlling the mosquitoes would block transmission, and this could lead to successes in program control, such as we saw in the Panama Canal. In the 1930s, Nobel Prize-winning discovery was that DDT was a fantastic insecticide, initially used for typhus, control of typhus, lice, and troops in World War II, but as it came forward and was tried against other insects, it was found that the effect against mosquitoes was instantaneous and very dramatic indeed. So it was so successful that in 1955, Russell, in his book Man's Mastery of Malaria, felt that DDT spraying and other control measures would make it economically feasible for nations to banish malaria completely from their borders. Started the 1955 World Health Eradication Campaign. Eradicate malaria, centrally controlled from Geneva, monolithic program, spraying and case detection and follow-up with availability of drugs, etc. So successful and so optimistic were the originators of this campaign that there was even a postcard celebrating the eradication of malaria from the Kennedy administration in 1962. India was a poster child for this initial success of this program. From millions of cases and epidemics in the early 1950s, malaria incidence plunged. By 1960s, there were just few, the number of reported cases in India were numbered in the thousands. So successful was the campaign that it did a better job of eradicating the malariologists and the control program than it did of eradicating malaria. As the political will and funding for spraying and other controls evaporated, and also there was a development of insecticide resistance by the insects themselves, malaria began to resurge, and by 1970s it was back to epidemics of malaria again in India. And the control programs since then have reduced it, but malaria is still embedded in areas of India today. So I touched on this earlier. I realize I have less time than I thought I did, so I'll just quickly say that with the introduction of chloroquine, there was that plunge in sub-Saharan Africa, and we know from our marker studies and all the boots on the ground epidemiology that as chloroquine resistance spread in Africa, that the cases resurged. Individually, we see this in our field sites in Africa, well documented in 1989 by Greenberg and his bulletin of the World Health Organization showed what happens when these chloroquine resistance strains entered a village in this area and what was Zaire at the time. In the Mama Yemo Hospital, the chloroquine-resistant strains were accounting for about 20% of pediatric deaths among the children. Chloroquine resistance entered that area, and within a year, they jumped. They doubled, more than doubled, up to almost 50% of the admission. So how did... by the late 60s and the 70s and into the 80s, it was clear that optimism, that World Health, that first malaria eradication campaign wasn't successful. It was abandoned in 1969. There was, and as I commented, much of malaria science has suffered a big knock during this period because of the optimism. When I entered malaria research in the 1980s, having experience in other areas of science, I looked around and I said, we have relatively few gray-haired eminences in this field now. They're gone. A lot of books, great books, published in the 1940s, 1950s, through the 60s and 70s. Not so many. I count on, the fingers, the number of real experts who stayed with the field and were available to teach and nurture us in that critical period. 1978, the World Health Assembly had recognized they needed to redefine from a centrally administered eradication program to a refined control strategy. And they also realized that phoning in the commands from Geneva for spraying and drug administration had to be changed. It had to be tailored to, the control had to be tailored to local epidemiological conditions and the resources available. And they called this stratification. Progressing further, I remember well in my earlier research career, the 1992 revised global malaria control strategy. Here we began to sense on part of health ministers across the world a greater cohesion and ideas of what needed to do to re-implement malaria control. And from that conference period, there came an emphasis on disease control that was based on decentralized, tailored use of anti-transmission measures and antimalarial treatments. Also, the importance, and here you're beginning to see the first emphasis on capacity and infrastructure strengthening, political commitment, long-term, very important message today, and community partnerships. The community partnership received very little emphasis in that first campaign. And I've listed others. I can't go into much detail because of time limitation. 1998, the big organizations established the Rollback Malaria Program. In 2008 was the convention of the Malaria Eradication Research Agenda. That was convened by the Gates Foundation. And Rollback Malaria released its first global malaria action plan. This year is version 2.0 of that action plan. So with reconsideration of malaria control in the wake of these major events in the past century, the malaria community today and the scientific research community recognizes the importance of integrated approaches in control programs. These are emphasized in the 2014 World Malaria Report, which I recommended highly for reading. And on the research side and the importance of good science in malaria control, the reading of the Malaria Eradication Research Agenda Initiative published by PLOS in 2011 is important reading as well. There are 4 pillars of research and control that emerge from these publications. Leading it, of course, is the availability of effective treatments for malaria everywhere. Mosquito transmission control means to control the mosquitoes not only by nets, but by other advanced measures as they become available. We recognize now the value of rapid diagnostic tools more than just thin blood smears that can be read. Oftentimes they're not read till days after they're taken, but these rapid tests that can be available and give you a readout from the back of a Land Rover or a cart within 5 minutes. And then eventually the value of malaria vaccines to control the infections and the severity of the disease, and particularly in the children. So let's comment a bit further, discuss a bit further the status of antimalarial medicines. Chloroquine, as we mentioned earlier, was a major drug of the 20th century. And its loss has led to a huge resurgence of malaria. The initial foci of resistance appeared in South America and Southeast Asia in the late 1950s. We recognize today from our molecular studies and from our discovery of the gene and the polymorphisms in this gene, it encodes a transporter of drug resistance. These polymorphisms serve as markers, and we recognize today how those foci arose and spread. Initially, the foci in South America, there were two of them we know of, swept south and covered the Amazon within a few years. And in Southeast Asia, in late 1957, there were initial foci that then spread village by village, country by country, throughout in both north and south. At the same time, accompanied by another focus that seems to have arisen in western India, spread up through Afghanistan and then across and met the Southeast Asian focus. Africa was spared for a time, about 20 years. We don't know why. It's an interesting question, what it is about the parasites in Africa that impeded this early entry of chloroquine resistance. It just wasn't there until 1970s. When it hit, though, it spread like wildfire. And across this vast region of sub-Saharan Africa, swept through those countries in a period of about 10 years. I've been discussing the major killer of parasite in sub-Saharan Africa. That's Plasmodium falciparum, responsible for about 90% of the deaths in the world today among the children. There is a second Plasmodium species, Plasmodium vivax, that also accounts for a tremendous public health burden in malarious regions. Not so much in Africa, where through the discovery of the Duffy receptor, and Lou Miller's pointing out years ago of the resistance that Duffy-negative individuals, particularly in Africa, have against Plasmodium vivax infection. Means that Plasmodium vivax is largely in South America and in Southeast Asia. We think it accounts for about 30 to 50% of malaria infections across the world, and its public health burden, when measured in disability-adjusted life years, or dailies, it approaches that of Plasmodium falciparum. And there's more and more recognition today of the severe morbidity and mortality that can occur, less frequently than with P. falciparum, but significantly with P. vivax. I forgot to mention also that P. vivax chloroquine resistance didn't emerge until the late 1980s, but now is spreading throughout Southeast Asia and throughout South America. So this, which really is a crisis in antimalarial drugs, you lose the frontline drug, and I don't have time to discuss the resistance to other antimalarial drugs that have been used through the 20th century. This crisis led the Institute of Medicine to convene a conference and committee, and to look at the quality, affordability, and access of drugs, and also what to do in the future in an era of antimicrobial resistance. This was published in 2004. A central recommendation from that study was that there be a sustained global subsidy of the more expensive drugs, particularly artemisinin. I'll come to those shortly, that have emerged in a co-formulated form as combination therapy and what are known as artemisinin combination therapy, or ACTs. Following that recommendation, and with other initiatives across the different malaria control programs, there's been a burgeoning use of ACTs for malaria treatment. But these are now encountering resistance as well. And here's a study published in New England Journal of Medicine in 2014 showing that one of the combinations, which had, and there was much hope for this one, dihydroartemisinin and piperacin, is now failing in Cambodia. Through population studies and an intense research effort by a number of laboratories globally, there's now a marker of delayed clearance after artemisinin treatment. There's no marker yet for piperacin failure, which is an important player in these failures. And again, the spread of these markers in Southeast Asia, Cambodia, Thailand, is just as intense as the markers to chloroquine resistance, for chloroquine resistance were 50 years ago. What do we do about it? Well, we need fundamental research, new drug discoveries, and the development and access of the new discoveries by the pharmacy and bringing them to the shelves where people need them. And this was a major question in the field in the 1990s. I mean, a lot of great ideas in academic labs, pharmaceutical companies with great resources, but they're not partnered, and there's no player in the middle to bring them together and say, hey, we really, and advocate for new antimalarial drugs. Well, public-private partnerships came to the fore, and they have been tremendous for malaria. As this Financial Times article pointed out in 2005, they work, they're cheap and effective, and they're the best outcome for public health. The Malaria for Medicines venture, conceived in the 1990s, has been a great success story for public-private partnerships. The portfolio today from these partnerships of academic labs with ideas for new drugs and pharmaceutical company with resources and the heart to take forward and contribute those resources to important drug discoveries for neglected or tropical diseases, has produced what probably is the largest portfolio of antimalarial drugs ever for discovery and development, antimalarial candidates. It's probably greater than the large program I didn't discuss during World War II, sponsored under the Office of Scientific Research, the same office that sponsored the Manhattan Project for screening and discovery of antimalarial drugs, which, by the way, was a public-private partnership that we recognize today for what it was, but at the time wasn't called that. And many of these drugs are the hope for the future as artemisinin resistance and resistance to other drugs begins to spread. What I'd like to mention, too, is that many of these discoveries have been found by this screening of malaria parasites for responses to large libraries of compounds, and the mechanism of action for some of these is not known. It's a screening technique. It's after circling around through structure-activity relationship discoveries and combinatorial medicine and several other things. There's a big emphasis today on screening natural compounds and compound libraries for drug discoveries, and so many have emerged from that. What do we attribute it to? A fundamental research discovery. It's possible because of what this fellow did, William Traeger, who died in 2005, member of the National Academy. He worked from the period from 1941 to 1976 to understand and develop a method to cultivate malaria parasites in vitro. Before that, it was really difficult to work. You had to look at malaria parasites in birds or in primates, expensive, and not always relevant to the human condition. And in one of his memorials, Larry Simpson and Erwin Sherwin wrote that the impact of this continuous cultivation of P. falciparum was phenomenal. A great inspiration for good fundamental science and the impact it can have in global public health. I mentioned earlier the P. vivax burden and its distribution. We have not found a way yet to grow P. vivax in the laboratory in continuous culture. We need it. Until we do, until we solve the problem, molecular and genetic progress on P. vivax and drug screening are going to be slow and limited. As Bill Traeger said, you can't study something you can't grow. A large amount of our work here at NIH with P. vivax has to be done in monkeys, and we really want to get away from that and be able to do it in vitro. Before I leave the antimalarial area, I'd like to mention that we also have a social problem and a political problem across the world with fake drugs. We can develop the finest drugs, but the counterfeiters and the poor quality manufacturers are creating an enormous problem, and it spreads drug resistance, not to mention the number of deaths that are left in the wake because of treatments that don't work. About a third of antimalarials are fake. And if you'd like to become involved or read further about this, I'd encourage you to go to the website Fake Medicines and Malaria. Read and look at the references therein. I mentioned malaria rapid diagnostic tests and their importance. These have emerged from our understanding, again, from fundamental research of antigens that are secreted by the malaria parasite and their ready detection by antibodies. They are formulated. Here we hijacked a test that's used for the streptococcal infections of the throats of children. And with the partnership, again, NIH pharmaceutical public-private partnership, if you will, found that these antigens could be used in that test to produce a pink band on a strip within a few minutes and demonstrate infections in the children. The use of these tests has expanded enormously in the period of artemisinin and combination therapies. In 2013, almost 300 million kits were produced, half of them based on this technology of the HRP2 antigen. This distribution has led for the first time in the recent years of more tests than ACTs distributed. Why is that important? Well, a couple of reasons. One is that in the chloroquine period, fever in Africa was equivalent to malaria, and every child who came to triage received chloroquine. I don't know how many children had pneumonia, had other infections. Chloroquine doesn't work against them, and those children were not effectively treated. But chloroquine is really cheap, and there was nothing else available. Now with diagnostics, we can know with pretty good certainty whether that is a malaria infection. ACTs are expensive. So an inexpensive, rapidly effective diagnostic test and appropriate drug therapy now are much better than the indiscriminate treatments of 50 years ago. And children with fever who are malaria negative move on to the appropriate therapy for their pneumonia or other infections. They aren't perfect. There's a large initiative centered in the World Health Organization to find those tests that have a great positive predictive power and specificity. On vaccines, I'd like to just spend a few minutes to mention that 2014 was a landmark year. The RTSS vaccine was announced in several articles and with editorials. PLOS Medicine carries one of the important articles showing that this vaccine is 30 to 50% effective among children in the 5 to 17 month age range for 18 months. It's not perfect. It's you know, an argument would be, well, it's just somewhat effective. That's true, and for perhaps a limited period of time. But when hundreds of thousands of children are perishing in Africa, 30 to 50% improvement falls well within that curve in the Millennium Goals to reduce morbidity and mortality further. Again, the lesson, fundamental research, long-term view has led to this vaccine. I'd just like to point out that it was spearheaded early on by Ruth Nussenzweig. Her publication in Nature in 1967 showed the initial protection by irradiated sporozoites injected in a mouse malaria model, carried forward and spearheaded by people, particularly Joe Cohen at GSK, the development of the RTSS vaccine, which, by the way, works because of it's for a couple of reasons. One is the understanding of of the epitopes in this vaccine, B-cell epitopes and T-cell epitopes, and also the linkage of this to the hepatitis B surface antigen. And this vaccine is great not only or useful not only for malaria control, but it's a great vaccine against hepatitis B. The public-private partnerships are active in vaccine development across the different phases of the malaria life cycle. What about the future? Well, many of the vaccine screening initiatives and work have been hit or miss to find those antigens that are recognized by sera that are in individuals who have immunity against malaria. But now we're approaching a new period where understanding of critical complexes in the malaria life cycle can be attacked. And I just illustrate a couple of these here. These for red cell invasion. The discovery of the essential role of the RH5 Ripper CYRPA, cysteine repeat protective antigen in malaria, produced by malaria parasites. They interact at the red cell surface, here shown in red with the complex basogen, connect to the parasite as the parasite invades the red cell. And if these can be interrupted by monoclonal antibodies or vaccine approaches, or perhaps even drugs, we may be able to reduce invasion and get good action against malaria infection. Also a discovery here at NIH, Prakash Srinivasan working on this with Lou Miller, is the AMA-RON complex. It comes, again, fundamental research long-term. The people working in toxoplasmosis found the importance of AMA-RON, John Boothroyd, for example, out at Stanford. And that work in toxoplasmosis, and what are you guys doing studying toxo-invasion of cells? Well, translates over to malaria. Research and discoveries work in ways that are difficult to anticipate, and we need to stay with it, and the advances will continue to emerge. We also need to understand more about malaria vectors. It's not just nets, and it's not just insecticide. There are more than 40, probably hundreds of Anopheles species, each with their malaria, their transmission characteristics, responsible for malaria worldwide, shown here in the different colors. And they don't all respond to the same measures of control. We know that the entomological inoculation rates are extremely important, and that we need to reduce them more than 99% to begin to make an impact on malaria infection in some of these high transmission areas. There are booms and busts of malaria during wet and dry cycles. What underlies that? What enables mosquitoes that during the dry season in Africa, where the land is parched and brown, to come back in a rush when the rains begin to fall? Where are they hidden? If we can find their niduses of expansion, if you will, their sources of expansion, and go after those areas, we might be able to make a dent on the big populations that occur in the wet season. And then here again, long-term research and the value, we don't know where it's going to lead. But Carolina Brias, who was spoke here a couple of years ago and was elected to the National Academy recently, has found the importance of plasmodium genes that mediate evasion of mosquito immunity. Have you thought about mosquito immune systems? You know, I've talked to people and they say, what, do mosquitoes have immune systems? Well, the answer is yes, they do. And there are themes of mosquito immunity that also play out in human immunity. Mosquitoes have ways of resisting infections, and the more we understand about that and the way the malaria parasites evade those immune mechanisms may lead us to unforeseen and practical measures of transmission control. So I'd just like to take a few minutes to conclude by coming back to the theme of under 5 child mortality by region and income. It's a disease of poverty. The malaria poverty cycle was emphasized by Winslow in 1951 in his World Health Organization series. He points out that men and women were sick because they were poor, and they became poorer because they were sick, and they became sicker because they were poorer. The breaking of that cycle is key. It's recognized as part of the International Bill of Rights, and in particular the International Covenant on Economic, Social, and Cultural Rights, the ICESR. This was, by all nations, commits parties to work toward economic, social, and cultural rights for individuals, including labor rights, rights to health, right to education, and right to adequate standard of living. If we consider the inalienable rights to life, liberty, and pursuit of happiness, this is a foundation, and it lies at the founding documents of our country. I, for one, am quite surprised that although Jimmy Carter, during his presidency, supported this, this has not passed through the Senate and Congress, with the idea that this contributes to global health care and is a laudable goal rather than a right. So if you'd like to read more about this, go to this Wikipedia entry and read about the history. It's still alive, and if we sign on in support of this and join other nations, it's going to be an advance for global health. Finally, just to point out socioeconomic development, published in The Lancet was this article in 2013, pointing out the importance of increased investments and interventions to support socioeconomic development, although they would not recommend discontinuation of existing malaria control efforts. I was really glad to hear that, or to read that, I should say. Now let's ask this question. Now and I'd just like to conclude with this. If we ask the question, is socioeconomic development going to do the job of malaria control, or do we need malaria control for socioeconomic development? Coming back to what I pointed out earlier, outside Saharan Africa, the tremendous drop in rates. This has been in China. Here you can see discovery of artemisinin compounds there, of course. On a log scale, malaria has dropped, there's tenfold, there's 100-fold. Here we are down almost 1,000-fold in the period 1970 to 2005. How did they do it? It's gone today from those areas of Canton and other areas where it was so rife. Well, if you read and see public health care nets, community participation, official commitment at all levels led the list. Others, the other related measures shown here. Scientific research and discovery was emphasized in China. Where's the economic development of China? Here's the boom, 1990, 2000, and forward. I don't think the temporal relation here can be construed as socioeconomic development leading to malaria control. Maybe we have the argument otherwise. There's been a global decrease of malaria transmission in areas of urbanization. This published in the Malaria Journal in 2012 and 13. And I'd just like to conclude with this short movie. This is by Hans Rosling. Visit his website. Take a look at this graphic in this series. He shows life expectancy in birth by years, which is a surrogate for health, income per person, which is a surrogate for socioeconomic development and wealth of nations. What has happened since 1809? And as I click on this movie, you're going to see the years go by. If health comes first, if wealth comes first, you're going to see the dots move this way and then health rise. And if health and control of these diseases come first, it'll rise this way and development will go, and then the wealth will proceed along the horizontal axis here. And I will click on this, start the movie, here it goes. You can see 1836, 44, some development. These are the countries of Europe and Americas. China, India here in the red and the blue, hanging back. 1903, 5, starting to, kind of can't tell yet. But now, 1940s, eras of vaccine, public health advances, where everybody's floating up, and now we're beginning to move to the right. By 2007, there it is. And he has an update from that. So, breaking the health poverty trap is going to involve health control. Just consider the size of Africa. I still contemplate this. China, United States, India, all these countries fitting in with Africa. And I'd like to leave you with a note of optimism. Despite what we read in the papers, every day you wake up to terrible headlines, right? The Middle East is exploding, Crimea is being invaded, people are shooting up schools. The statistics globally are impressive, and there's fair arguments. This Think Progress sites point out 2013 was the best year in human history, measured by measures of death and war and extreme poverty and murder, etc. And statistics for 2014 have looked even better. So I'll conclude with that. Thank you for your time. I just look at the paper and then read it.
Great. Okay. Everyone hear me? Well, thank you for that global picture. I'm going to take a deep dive into the human organism now, thinking about the parasite as it goes through its life cycle in the human, and thinking about what kinds of new tools we might have to offer the anti-malarial and vaccine development efforts that Tom so eloquently spoke about. And I'm going to just my talk will be in three parts. The first third of the talk will actually be talking about the preamble to how we got into malaria, which is we were trying to build artificial human livers one day for transplantation into patients. And I'll show you that engineering tools that have come out of the computer chip industry have been very relevant for this area of tissue engineering or regenerative medicine. And the second two parts of my talk will be focusing on tools that we developed for other applications and using them for falciparum and Vivax applications. Okay, so as Wynn mentioned, I'm an engineer by training, and my specialization is in miniaturization. And so in microfabrication and nanotechnology tools. And so this is an image in my world that really motivates us in terms of the kind of progress that has happened. And you may know this visual. So this is a single transistor 50 years ago. And now actually you can fit close to 1 billion transistors on the same footprint. Okay? And this is the power of team science, engineering, physics, material science working together. And really what drove this, of course, is an interest in speeding computation, which has changed all of our lives. Everyone in the room who has a smartphone has one of these chips inside it. Really at the core of this whole miniaturization revolution is a very simple process. It's called photolithography. And the way this process works is you take a surface, in this case silicon, you coat it with a polymer that's light sensitive, you shine light on it, that's photo exposure of that, and you develop the exposed regions like film. And what that allows you then to do is to draw circuits on these chips. Okay, and the reason that we can now fit so many more of these is that the resolution of this technique has gotten better. So it used to be in the 70s that the line width that you could make was 10 microns, and now it's a few hundred nanometers. Okay, and so this photolithographic tool has gotten better and better over 50 years. So what we've been interested in now, thinking about this from a medical perspective, is not just this one tool, photolithography, but a whole suite of tools that have emerged from this community of engineers that are very good at manipulating objects at the 10 micron and under length scale. What's kind of interesting about that when you think about the human body, so this is my favorite organ, the liver. They look up a blow up of the sort of the environment of hepatocyte in the liver, and you think about all the things that this hepatocyte experiences in its native microenvironment. Well, a single receptor on the cell is order 10 nanometers, and multiple cells next to each other are order 10 microns. Okay, so we have a set of tools now to control cellular organization, cellular microenvironments with exquisite precision. And so the question is, what could you do using these engineering tools in recreating microenvironments and studying these microenvironments? Okay. So what I'm going to tell you about is first a problem in the liver field, which is that when we take hepatocytes out of this native microenvironment and we try to use them for some practical application, for example, like supporting a patient in liver failure, one challenge is that this hepatocyte now in culture will very rapidly lose its repertoire of functions that it had in vivo. This is just one function, which is the production of albumin, but sort of take your favorite liver function, coagulation factors, production of bile, all the functions that the liver does will very rapidly decline under sort of conventional culture conditions. Okay, and so what we have been interested in our group are three things. The first is trying to understand this hepatocyte microenvironment. What is it about the microenvironment in vivo that's now missing in culture so that we can understand sort of the structure-function relationship of this tissue? The second thing is then ultimately applying this to creating an artificial liver that you would put in a patient. The third thing, which is the thing I'll focus on today, is if you can make a little human liver in a dish, can you model human diseases? Can you study, for example, liver infections for which there are not good animal models of disease? Okay, so when there is actually a long, long history of knowing how to culture hepatocytes in the field, and I don't want to give you the misimpression that we don't know anything about this. So people have tried different media compositions. They've tried to vary the extracellular matrix that the cells saw in vivo and putting them in vitro. They've tried to culture hepatocytes together with other cells from the liver. They've tried to grow them out spontaneously over extended culture. All kinds of techniques have been tried. But one thing that really intrigued me when I got started in the field was this observation, which was made in 1983 by a liver researcher named Christiane Gugen-Ghiozzo in Paris. What her group showed was that if you took hepatocytes and co-cultured them with non-parenchymal cells from the liver, at the time she was using another cell that she thought was a biliary cell. So she co-cultured these two cell types together, you could get some rescue of the phenotype. The cells weren't organized, and the rescue wasn't forever. And she hadn't at the time done it with human cells, but nevertheless it was a very powerful effect. So as a graduate student, knowing that microfabrication was this really powerful tool, we thought, well, maybe it would be interesting to try and organize the cells. So if we're co-culturing them, so this was her classic experiment, now I've just color-coded it. So red are the hepatocytes and green are the supportive non-parenchymal cells. This is what we call a random co-culture. what I decided to do as a graduate student was to take photolithography, which is the technique I described to you earlier for patterning silicon surfaces, but now just to pattern cover slips. So take a cover slip, pattern the cover slip with collagen, which I knew the hepatocytes would stick to, and now we could create what we call micro-pattern co-cultures. And using this simple manipulation, now I could study the architecture of this tissue and how that affected the function. And what I found was that just taking the same numbers of cells and repositioning them on a two-dimensional surface could dramatically affect the rescue of the phenotype and the persistence of that rescue. Then we got interested in the dynamics of this. So a postdoc in my group created this little chip. This is a piece of silicon, so it's actually crystalline silicon, and he's coated it with polystyrene, so it looks like a tissue culture surface. And he made this part, we call these the interlocking combs for obvious reasons, so that we could bring the cells together and apart. And we can ask questions not just about what the architecture of the tissue was, but also the dynamics of that process. Do they have to touch? How long do they have to touch? Can they share soluble factors? Okay. So over the course of 15 years, we learned a lot of things, and I'm not going to go much into it today because I want to talk about malaria. But I'll just say that we learned that this co-culture effect that was first reported in the early 1980s had two temporal timescales. So you had to touch your neighbors for a day. There needed to be a contact signal. And after that, you could be 400 microns away, and you could share soluble signals. Okay? And that seemed to, so this co-culture effect had these two components, and that the nature of your neighbor mattered, right? It's just like in real life, the neighbors matter. And actually, you could get some level of support from lots of different non-parenchymal populations. So the first reports in the 80s were with biliary epithelial cells. What we found, there's serendipity in science, what I found was that the cell line that I stumbled upon, which was first cloned by Howard Green at Harvard Medical School, because it supported keratinocytes very well, and later we found that out because it made keratinocyte growth factor. This particular subclone of fibroblasts that he had seemed to be the best in our hands to date for supporting hepatocytes. It's not actually obvious why that would be. They're a mouse embryonic fibroblast subclone. We've tried 30 different cell lines, and they just happen to be the best. So we obviously are doing all kinds of transcriptomics to find out what's so special about them. And we have sort of some various theories at the moment. But so far, we've found things that they make. So they have special cadherins. They make becrin, a particular extracellular matrix molecule, and they make some soluble factors that seem to be involved with this, but we can't replace them. So they said, okay, for now, we're engineers. We have this sort of stable culture system. What could we do with it? Okay. So a graduate student in my group, Salman Kitani, who Wynn knows well, decided to apply this to human hepatocytes. So what he did was take these micropattern co-cultures, vary the size of the colonies, vary the spacing, vary the ratio. And what he was able to achieve, just by empirical optimization, was this stability of human hepatocytes over the course of about 4 to 6 weeks. So here I'm just showing you again this albumin biomarker, but this is true for a whole repertoire of functions. And one of my favorite kind of visual examples of this is if you give these cells an analog of fluorescein, which they secrete through MRP2, which is a transporter, at the biliary canaliculus when the cells are polarized, you can see the biocanalicular tree light up. So for those of you who aren't used to looking at pictures of the liver, if you look in an anatomy textbook, the biliary network is like a chicken wire diagram between hepatocytes. And what was really surprising to us is that we're seeing this recapitulated in two-dimensional culture kind of en face, right, in a culture dish. So it's not necessarily what we were expected, but we were super excited. Okay, so then the question is, what would you do with the human liver? So one of my students in the lab is actually really interested in art. So he's collaborated with this brilliant Brazilian scientist, artist, Vic Muniz. And this is actually hepatocyte wallpaper. This is part of a colonies exhibit. It's traveled the world. This has been in New York, in Tel Aviv, and in Brazil. And they've just been micropatterning beautiful structures that have this sort of collective property. So you can see some more of them here, which personally I think are gorgeous. But a very practical thing to do is to make a drug screening platform. Okay, so then what we did was to say, okay, if we have little human microlivers, what if we make them in multi-well plates so that as a user, you could take off the shelf a 24-well plate or a 96-well plate, and it fits into your spectrophotometer and your robotic fluid handler, and then you could use it just like you would use any cell culture. It just so happens in each of these wells are micropattern co-cultures of the optimal size. So that's what we did, and in order to sort of really get this working, we spun a company out of MIT called Heprogen, which now markets this to pharmaceutical companies for drug metabolism and toxicity testing. I mean, you heard a little bit about drug metabolism in the context of antimalarials earlier. It turns out that many animals have different drug metabolism than humans. And this is well known. It's mostly at the level of the P450 family of enzymes. So one thing we wanted to see was whether we could predict some of those things. These were some of our kind of most exciting results. This is a toxicity assay with a drug known as phyluridine. So phyluridine was actually a hepatitis B drug in the mid-90s, which sadly caused 5 out of 15 lethalities in a phase 1 clinical trial. And this is in spite of the fact that the drug had been studied in vitro, had been studied in small and large animal models. So sort of unexpected toxicity. And what we see is if you put it on a human microliver culture, the toxicity just really jumps out at you. So it's very species specific and dose dependent. So the way it sort of works at MIT, at least, and I'm pretty sure at the NIH, is once you start a company, you can't really work on those problems anymore, right? Because that's a conflict of interest. You and your students should do something else. So we thought, okay, what else would we do if we had a human liver in a dish? What would you do? So one thing you might think about are all of the liver infections that are specific to human hepatocytes. So there's 280 plus cell types in your body, and this class of organisms only infects the hepatocyte and only infects primarily the human hepatocyte, maybe some primate hepatocytes. And actually there's several organisms like this. So hepatitis B virus, hepatitis C virus, and malaria. So falciparum and vivax actually stop in the hepatocyte on the way to the blood, and that's the place they go first. So we started first working with Charlie Rice at the Rockefeller, who Wynn Arias introduced us to, and infected the cultures with hepatitis C. And we infected a whole bunch of those other ones that I showed you earlier, the matrix configurations and the different media. And what we found was that in the micropattern co-culture format, you could get robust HCV infection and drug sensitivity. And then in 2008, as Tom showed, because of the sort of renewed call to malaria eradication, I got invited. This paper had just come out describing the drug platform. I got invited to the Gates Foundation because they were deciding that we needed to sort of have new tools for early stage research on the liver. And I actually didn't know much about the malaria problem at the time. I kind of knew that malaria infected the liver, but I wasn't sure what the right questions would be. And certainly, since it's not endemic in the US, we had no access to any reagents. So you really needed somebody to, you need to sort of be invited to the party. So I got my party invitation in 2008 and learned about the global burden, which you've heard about, which is driven by falciparum and vivax. And Tom has already told you how much we need a better vaccine and that drug resistance is emerging. So I'm a visual person. I think movies are helpful. So this is just for me to describe how the parasite gets into the body. First actually goes into the hepatocyte, expands about 10,000 times, so it amplifies. This happens over the course of a week. Then it bursts out of the liver. Then it infects the blood. And then we get the cycles of fever that you heard about. These, then a naive mosquito can sample this blood, and then that's transmission. Okay? And so because of this recognition about the liver stage, it's a very attractive place to target the parasite. So why is that? Because first of all, this is a site where there's less bioburden, right? If you can get it before it amplifies 10,000 times, you have less parasite to kill. It's an asymptomatic stage, right? There's no disease yet. And if you can block it here, then you would block the disease and potentially also transmission, right? And so for this is the reason that people were interested in liver stage targets. There are a couple of antimalarial drugs that target the liver, and I'll show you that later, but they're pretty old drugs. They need to be improved. The other interesting thing is that the liver has also been a site for vaccine development. This is what they call the pre-erythrocytic vaccine. So typically what's done, and I think Tom, you mentioned this, is you could take a parasite, attenuate it by irradiation or now genetically, okay, so it can't cause disease. But in order for these parasites to effectively raise an immune response, they typically go into the liver, set up shop, express antigens that are liver stage antigens, raise an immune response, and then they arrest there in the liver. So in order to study that, in order to study your genetic attenuations or your irradiated parasite, again, you need sort of a human liver stage model. And then obviously if you have both the host and the pathogen, then you could study these interactions. Okay, so obviously people have been working on this for a long time, and there are existing models. So the first one is a hepatoma-derived cell line. There's a whole family of these that are widely used, and they actually can be infected. They're permissive. The problem is that the host that the parasite is growing in is a cancer cell. So it's a cancer cell, it's dividing. It's pretty much the opposite of a quiescent hepatocyte that hosts something for a week, right? They have deficient drug metabolism. So there's lots of problems with those cells. I mean, they have been fruitful, but I think especially for Vivax, which is, as I'll show you in a second, has this hibernating form, which hibernates in the liver for weeks, months, years. It seemed reasonable to us that you wouldn't want a cell that was dividing every 24 hours. So one kind of encouraging thing was that another French researcher, Dominique Mazier, around the same time that the initial co-culture experiments were done, 1983, she had published 2 back-to-back amazing papers showing that human hepatocytes could actually support the whole liver life cycle of falciparum and Vivax. These are really seminal papers. So when I came into the field, we saw these papers. I actually met Dominique at this 2008 meeting. I asked her, why aren't people doing this? Because this seems like the authentic host. And she said, it's just technical. There's no reason. It's just that I'm sitting there and I have an operating room with fresh hepatocytes and I have an insectarium with falciparum and I can do the experiment. But no one else basically in the world could do that experiment. So if you're an engineer, that's actually music to your ears, right? Because that means it's an engineerable problem. And so we set out to see if we could engineer a kind of more tractable solution. So the first thing that we did was start to use different hepatocyte donors in our micropattern co-culture system. Because in the intervening years, one thing that happened is that the pharmaceutical industry had figured out how to freeze hepatocytes. So the cryopreservation of hepatocytes had come into play. And this was sort of, again, serendipitous. It was not our research, but we benefited from it because what it meant is that we could now go to a freezer, try 30 different livers, and if one of them was permissive, we could literally procure the whole lot, 300 vials, 1 billion cells, and do our experiments for a year or two. And so that's essentially what we did. So we started screening donors, and what was so interesting about it is that we could get, so you can see here these 8 donors, they all patterned beautifully. They're relatively functional by many different markers of hepatocyte function, but their permissiveness, the relative infection to falciparum is actually really variable. And we couldn't find biomarkers of this infectibility. For example, one of the entry receptors for falciparum is CD81. And you can see that many of the ones that are infected also express CD81, but some of them have low levels of CD81 and they're also infected. So that was not a good enough biomarker. So there's some host variability that we don't understand, and we sort of reason that this has been one of the challenges in the field. If you're just taking whatever liver comes out of the OR, some days you'll get an infection, some days you won't get an infection, right? So how are you ever going to do a drug screen in that setting? So we pick our favorite donors, we buy the whole lot, and then we do experiments for a year. Okay, so 5 years later, this paper came out in 2013. We worked with several malaria researchers. We started working with Stephen Hoffman at Scenaria, and then investigators at Hopkins, and then Maria Mota at Lisbon. The field has been really welcoming. Stefan Kappa at SBRI, kind of Potini Sinis, many others. And so now we are able to show that the cells have the entry receptor, get parasites. This is what they look like. It's actually kind of, I don't know if you've ever seen one, but I think when I first saw this, I thought they were really creepy. You know, that these are actually swimming in your blood, sort of 1 micron long, on your way to the liver to set up shop. We can see them traversing hepatocytes. So this is actually a wound healing assay where the parasite traverses many other hepatocytes before picking one to set up shop in. You can stain them before and after they've entered to see how many get in. And if you watch them over time and you look for late-stage markers, you can see that these are called extraerythrocytic forms, EEFs. And you can see them growing here, multiplying about 10,000 times inside a hepatocyte. This is actually the hepatocyte nucleus, just to give you a sense of scale. So they've grown enormously. And then finally, if we take one of these cultures and we overlay human blood, so for falciparum, this would just be erythrocytes, we get bursting into the blood stage. So we have what we think is a full life cycle. And so you can quantify this now because you're on a drug screening platform and you can see these forms growing over time. And there's variability in both the parasite, which there's batch-to-batch variability when you get them from different mosquitoes. And we've taken the host variability away. And so now we can basically get very consistent infections of the kind that you could potentially do anti-malarial work on. We automated this with colleagues from the Broad Institute so that the students didn't have to count these little red dots. And now we have drug killing curves. So these are the two known liver stage anti-malarials that exist. Primaquine is a pretty old drug, and atovaquone, which is a newer, more potent drug. And you can see that they both have nice IC50s in this assay. So we think that it's a tool that you could use for new candidate anti-malarials, potentially. One really interesting thing about primaquine is that if you compare, I told you that the parasite can grow in some of those growing cell lines. So if you compare it to the HCO4s, which are a hepatoma cell line that were developed in Thailand, they also generate a kill curve, but the drug is actually much less potent. And actually, what's known about primaquine is that it's bioactivated by the P450s in a hepatocyte. And since the hepatoma cells have lower levels of P450s, we reason that this would have something to do with that. In fact, if you look at a couple of the known bioactivators of this drug, you can see that the cell lines have a lot less of it, and that the primaries have more of it, and you would kind of predict that the drug would be more potent in a primary. So it remains to be seen if this is actually useful, because nowadays with chemical biology, you wouldn't want to make a drug that was bioactivated. Usually they try and design that out at the medicinal chemistry level. But nonetheless, I think that the primaries are more predictive for things that rely on liver metabolism. And then finally, just sort of like one tidbit of a new thing that we found. So what I told you so far is that we can do sort of clinical drug metabolism in this little mini liver. One thing that we got interested in was that the seminal breakthrough that Tom mentioned. So in 1976, continuous blood culture of falciparum was enabled. And actually the story of that, you probably have I only know the written story, but the story that I've heard about it had to do with a candle jar and hypoxia. And one piece of the puzzle was low oxygen tension, culturing the parasite at a relatively hypoxic environment. No? Yes? Oh, interesting. So it's CO2 and oxygen, or just CO2? Yeah. Okay, well, so maybe I'm happy that I got the story wrong, because what I was interested in, just coupled. So what intrigued me about the story, the oxygen part of the story, was that in the liver there's a gradient of oxygen tension, and we know this from the periportal region to the perivenous region, but actually everybody does the liver stage culture at 21% O2. Which is what we all do our culture in, right? That's what typical cell culture is. So we thought, well, what if we vary the cell surface oxygen tension in these cultures? And since we micropattern them, there are several ways to do it, but a trivial way to do it is to just pattern more islands in each well, right? So as an engineer, if you increase the consumption rate of something, then you're decreasing the local oxygen concentration. You can model that. But this is a visual example of something called a hypoxy probe, which stain cells that are hypoxic, and you can see here basically that we're getting different cell surface oxygen tensions when we pattern the islands closer and closer together. So now the question is simply, how permissive are these cultures at different oxygen tensions? And what we found was that there was a huge dependence on ambient O2. So if you look at predicted cell surface oxygen tension, actually, there's a huge peak about a log lower. So actually, we normally culture here 110 millimeters of mercury. So we're a log and a half lower. That's where our peak is. And we can show that this is driven by hypoxia-inducible factor, which is maybe the first thing that we guessed. And in fact, if you even just culture at ambient oxygen and use a hypoxia-inducible factor inhibitor drug, the DMOG, which impinges on this pathway, you can mimic the same effect. Okay. So anyway, so this is sort of learning about the environment of the hepatocyte and the host, and learning that maybe culture conditions would be better at lower oxygen tension, which is what the parasite probably also experiences in vivo. Okay, so that was all that work done with falciparum, which Tom mentioned is the bigger killer. But I think one of the reasons that I got really intrigued when I went to that 2008 meeting, one of the sort of grander challenges was this organism, the vivax, right? And so vivax, as was mentioned, is a barrier to eradication. So it impacts a smaller number of patients, but has some very interesting biology. So what's thought, and this is, I would say, still emerging. What's thought to happen now is that the parasite, a bolus of parasites come into the liver, and there are thought to be potentially two subpopulations. One population, which goes on to grow into these EEFs, just like I showed you before, but another population that forms what people call hypnozoites or sleeping forms because they're thought to be hypnotized. And these guys can hang out in the liver for weeks to months to years, okay? And at some later state, they reactivate, go through this maturation, burst into the blood, and give rise to what we call relapse, okay? And so what excited me as an engineer about this is that the time scale of this, the quickest possible relapse, is about 3 weeks, 3 to 4 weeks, which means you need a culture system, if you want to study this, that at least lasts 3 to 4 weeks. And our culture system lasted 4 to 6 weeks. So we thought, okay, maybe the time scales are lining up to be able to grow this and study it and find drugs that could kill it. Okay, so some sort of clinical data, epidemiological data to suggest that this is important, is this study, which came out in Lancet in the year 2000, where they tried to do eradication in the South Pacific in this little tiny island. And what's interesting about this is that before they did the eradication efforts with insect-treated bed nets and drugs, was that there was more PF on the island than PV. And after a year, all the PF was gone. And it took 5 years to get rid of the PV, right? And it's because of this hypnozoite, this sleeping form, where you can basically get relapse. Once you have relapse in the blood, you can reinfect the mosquitoes. You start the whole cycle again, right? So it's really a barrier. There's only one drug right now, primaquine, which kills the hypnozoite. We have this beautiful history of malaria research. Actually, this is a relative newcomer. So these are some glimpses of the hypnozoite from 1982, which is pretty recent. I was in high school. You can see these are images from chimps, that liver biopsies from chimps, where you can see just little glimpses of by immunofluorescence and heme sustaining of what these forms look like in the liver. And so we were really interested to see if we infected our livers with vivax, would we see these small forms? Would they persist? And then would they reactivate? So this is animated funny. So these experiments are really hard. In order to get these data, my team waited for the rainy season in Thailand. Patients came into our collaborators' clinic that had vivax. They harvest their blood, feed the mosquitoes, dissect the salivary glands of the mosquitoes, get the parasite. We make the livers on site in Thailand, then infect them, fix them at different days, ship these cultures back to Boston, stain them to see if they're infected, okay? And if it doesn't work, you wait for another rainy season, right? So we've done this experiment 4 times. This is, I think, the 2nd time. But so what you see is, so here on the right-hand side, you see infections. These are EEFs that are growing over the course of a week. And if you overlay, now this is actually harder. You can't just overlay erythrocytes. You have to overlay very young blood cells called reticulocytes, which is what P5X infects. But you can see burst through into the blood. So we have the whole growing life cycle. And then you also see these guys. And you can see that they persist, these little sub-5 micron positive staining organisms in the culture. Okay, so we started showing this data in meetings a couple years ago. We said, like, okay, this is suggestive of a hypnozoite. Please tell us how we could prove that we have a hypnozoite. Is there a marker? Is there some EM structure? What could we look at? So first of all, people said, well, there's no known marker because the transcriptome's not known. So we don't know any markers. The only two things that you could do to prove that it's a hypnozoite would be to show differential drug sensitivity to the one drug, primaquine, which is known to kill it, versus one that's not known to kill it, atovaquone, or see if you can wake it up. And so, okay, how can I wake it up? Well, we don't really know. But we could infer from clinical data. So from those same sort of World War II from wartime, not experiments of man, what we see is that actually patients that have hibernating vivax, if they get reinfected with falciparum, their vivax will actually flare. So there's sort of interesting data about vivax flares that you can infer from the clinical literature that you could sort of try to mimic in culture. Okay, so we tried to do both of those things. So this is the first thing, where we're trying to do differential drug sensitivity. So we have these cultures, the vivax-infected livers, and they're growing. This is the sort of spread of EEF sizes. If we treat them with this drug, atovaquone is only supposed to kill the ones that are growing, okay, not the small forms. And if we treat it with primaquine, we get fewer of those, and we get some small forms hanging on. At day 21, this is what we see. So at atovaquone, there's still some small forms there. So you might reason that makes sense, right? The large ones have been killed and the small forms are hanging on. They could be hypnozoites. And in primaquine, there's nothing left at all. That makes sense. It's supposed to kill both of them. And this was an accident, that an absolutely thrilling, like, best moment in the last 5 years. In this culture, what do you see? This guy, so after 21 days, in these cultures that have been cleared of the small forms, these guys look about 8 days old. That's how big an 8-day-old EEF is, suggesting that they've reactivated, right? Because normally we only see big forms at day 8, right? So this culture has been treated with atovaquone. Everything that's left is supposed to be small, and yet there's a couple of these big guys cropping up. So there are signs, there are reports in the literature for cynomology, which is a primate malaria, that you can get spontaneous activation in culture. And that had been reported last year. And so this is now we're starting to do immunofluorescence of these to see what they look like. And they look crazy. Like we only have 2 of these in the microscope so far. They're just ginormous. In the lab, we call this Gigantica because they wanted to name her. And anyway, so it's early days, but we think we might have spontaneous reactivation and differential drug sensitivity. We obviously have to do this more times, but I won't know for another year if it works again. Okay, so I know the hour is getting late. Just to summarize this part, so we've done these engineering tricks, created these human livers, and tried to model the liver stages of these 2 parasites, and tried to create a platform that the community could use for, studying new anti-malarials. One thing we are starting to do is work with organizations like MMV. So they send us their drugs that have blood stage activity. This is a drug they sent us, for example, over the summer that was about to go into clinical trials to ask whether it had liver stage activity, because that can inform the clinical regimen that they design, if it would have prophylactic activity versus blood stage activity. And then we're also starting to work on liver stage vaccines. So Scenaria, who is one of the earliest partners that we had, is actually a vaccine company. So they have an irradiated parasite. They actually grow the mosquito sterilely. They irradiate the mosquito, and they sterilely harvest the parasite from the salivary gland of the mosquito. That is their vaccine product. So they have this scaled up in GMP manufacturing. And the story with that vaccine is that it should infect the liver, right? But it should not burst out into the blood stage. Otherwise, it would be causing disease. So in fact, that's what we see. So we see EEFs, and they're attenuated as compared to the wild-type parasite, but they don't grow. They don't grow very Another very interesting thing was actually David Fidduck at Columbia came to us, and he had a genetically attenuated vaccine. And they had characterized it previously in that HCO4 in the cell line that I told you about, the hepatoma cell line, and it seemed like it caused arrest. But there had been some issues in the vaccine world where vaccines that are genetically attenuated had been put into patients and then had what they call blood stage breakthrough. So the community had been pretty gun-shy, and when he heard about our assay system, he wanted to see whether the parasite was arrested in our system or not. And as you can see, it's completely not arrested. So this is really a different answer when you use the prime hepatocyte than when you use the hepatoma. And so he was actually, we were really nervous to tell him about this result, but he was wonderfully embracing of it. And so the paper, which is now in press, says, this gene does not arrest at the liver stage. And then finally, we think that you can use this assay system potentially to query clinical data. So if you immunize a population with a vaccine and you have antisera from those patients, one question you could ask is, how good can you make a test that tests the antisera in vitro? And the reason you might want to do that is right now the way the clinical trials are done is you re-challenge the patients. They call this challenge. So you take a patient who's been immunized, you give them drug-sensitive malaria, and you see if they get sick or not. If they get sick, then you give them the drug. And so these are paid volunteers. That's how the clinical trials are done. So we thought, okay, what if we could just take the antisera and study liver stage activity in this assay? We actually didn't expect it to work because there's a lot of data to suggest that T cell killing is really important in the vaccine. But nevertheless, we worked with this set of data from patients that had that irradiated vaccine that I told you about. And then essentially what we see is that the patients that are protected clinically do score in this assay in a liver stage infection assay. So there's something in the repertoire of antibodies that is impacting establishment of the liver stage in our assay. So whether it's going to be a perfect predictor or not, I don't know, but you could start to ask lots of interesting questions. For example, which antigens on the parasite surface are driving the protective immunity in this model. So it's early days for applications to vaccine development, but we're kind of excited about that. So I think I'm going to skip, what do you think, Gwen? Second half? Yeah, because I think we should have time for questions. So I'm going to skip all this, just to say that we're making these little livers now in mice, too, in case you want a mouse that gets human malaria. And this mouse actually has malaria. This is a fake liver. It's a 3D liver that we put in the mouse belly and infected with malaria. So now we have kind of an in vivo model of the human malaria. In Burgii in this one, and Falciparum in the next one. Yeah. Yeah, the human cells are infectable with Burgii, Yoelii, Falciparum, and Vivax. Yeah. And then finally, just to finish up, one thing I think going forward, which has been an exciting kind of advance in the field, is the idea that there are, rare clinical observations that you want to study in terms of mechanism. And now we have a sort of new tool for doing that, which is IPS reprogramming. So if you have patients out there that have an interesting genotype with regard to malaria, you could take a fibroblast punch biopsy, take their fibroblasts, reprogram them with Yamanaka factors, and then do directed differentiation into hepatocytes, and then you have human hepatocytes from that person. And then you can infect them with parasites and study them. So I'm not going to show you, but we just published a paper last week showing that we can infect human-derived iPS cells that have been made into hepatocytes with Falciparum and Vivax. So it's kind of an exciting time. I actually am really curious about whether it turns out people don't really know if there are liver stage phenotypes out there. If you ask clinical malariologists, are there any interesting rare genotypes of the liver in terms of their malaria observations, because it's a clinically silent phase. I haven't yet heard sort of a genotype that's been nominated. Lots of people have interesting blood stage genotypes that should be nominated. But I think this will be interesting to see, what liver genotypes have different host factors for a parasite infection. So just to sort of sum up, we've been using these tools for studying the parasite microenvironment, for modeling human diseases, and hopefully for one day making these transplants. We actually do have some new data on this that I'm happy to share for at the end of in the discussion time. And even if you're not interested in the liver, hopefully you can appreciate that I think some of these tools could be useful for whatever your favorite tissue is, for modeling disease, for monitoring disease, for treating disease. So thanks for your attention. This is a fantastic group and funding sources and collaborators. And I think Tom and I would be happy to take questions.
Thank you very much. Thank you too. So I'm sure there must be some questions or comments. Anybody, please speak up. Try and keep it briefly. Yeah.
Hi, great talk. I was wondering about your results with the oxygen levels. Were you able to replicate those not using kind of engineered techniques for controlling the density of the cells of the co-culture, but just looking at, putting them in a hypoxic incubator and seeing whether you could see the same results?
Yeah, that's a great question. So the curve actually that I marched out where we looked at cell surface oxygen tension, we did that by varying ambient oxygen, and as well as varying the concentration of the pattern, as well as varying the height of the media, which is the diffusion barrier, is a third variable. So all three ways recapitulate that same effect.
Yeah.
You said you had done a number of 2D cultures. Had you also built 3D models?
We have. That was what I skipped. The thing that went into the mouse is a 3D version. It's basically a porous, like a sponge-like material. And then when you see that with hepatocytes, they form little aggregates. Yeah.
And at what size levels are you doing that?
So we call them our soft contact lens livers right now. So that's about how big they are.
And the line widths for the...
So in that version, those are randomly organized. We have one that's patterned by 3D printing. And those are still only about 1 millimeter thick right now. The reason is that the liver actually can regenerate. And so our hope is that we don't have to make them very big. We can make a small one, and then we can encourage the hepatocytes to grow. And then they will expand in situ. And actually, we've gone off screen, but that's the thing that just got working over the summer. So we have them growing 50-fold right now.
Hello?
Get vascularized?
They do. So we put them in together with, I'll show you at dinner maybe. we put them in together with an endothelial tree, and they seem to, it's kind of known that there's a paracrine signaling loop between the hepatocytes and the endothelial cells. And so what you see in the structure at the end is an integrated hepatic endothelial tree, and you can see erythrocytes in the endothelium, so they also seem to be perfused.
Do you want to add? Oh, good. Go ahead.
Hi. About the hypnozoites, could you isolate them and try to study only them, like extracting RNA or protein or anything else? Yes,
that's exactly what we're doing. So now, so we thought about doing it 3 ways. So the first is laser capture microdissection, but the problem is there's no reporter parasite, right? So because if you could see, with HCV we've done that, if you can see which cells are infected, it's like less than 1%, then you could just cut them out with laser capture microdissection and sequence them. So since that won't work, the new thing that we're trying is one of these single-cell analysis techniques where basically you disrupt your culture, you single-cell sort into FACS, you RNA-seq everything, and you just score the cells that were infected.
You try to do a backflip like a marker, like HSP7.
Yeah. HSP70 was reacting. Can you use as a marker for FACS and like sorting the hypnozoite cells and use it like for this analysis?
It would have to be a live cell immunofluorescent assay, right? So I think if we just do it blind and just do it by RNA, it may work without having to do the label. The other thing we're doing is just enriching. There's ways to enrich the parasite DNA and RNA from human genomic DNA with some baits, so we're trying that as well. But so hopefully we can have a transcriptome of the organism, which would be amazing.
So the hypozoites surrounded by a membrane?
Yes, they have this parasitiferous vacuole.
So what is the membrane?
So I'm not an expert in this. Tom probably knows better, but it seems to have some docking mechanism with the autophagy pathway.
Right. So it's like the parasitiferous vacuole of Leishmania and things like that?
I don't know about leishmania, but I guess so.
What, do you know what the pH is within that? Because all of those other membranes have a proton pump and they're acidifying. And that's the basis for like leishmania or leprosy or something like that sitting there for years until something happens immunologically and it bursts forth.
Huh. Tom, do you know? Is there a proton pump in the parasporous vacuole?
Not well characterized. Well,
but I think that would be interesting to do some comparative biology of other persistent organisms. There is apparently in the annotated genome, there's apparently a region of the genome that's been annotated as dormancy-like in the sense that TB, in relationship to TB. I have a question for you. Are we allowed to ask each other questions?
yeah.
What led to chloroquine resistance? What was the...
Here.
Question, what led to chloroquine resistance?
Misuse?
The amount of chloroquine use approached hundreds of tons per year. I mean, think of the hundreds of millions of doses. And each child, has, 10 billion or more, 100 billion parasites in the bloodstream. So
it's just incredible drug pressure.
Yeah, so if you think of the number of children and the number of parasites in each child, very infrequent events become very probable. And those events are point mutations in the genome. When they arise, and we know there's a spontaneous rate of occurrence of mutations going on all the time, they were very rapidly selected. And they come forward, and then, of course, with the efficiency of mosquito transmission, it goes from child to child. You can have a whole village, and it's been shown several times how quickly those drug-resistance organisms can spread. It's in our, in the course of just a few years, well within our lifetime, we're seeing Darwinian evolution in action.
Jerry?
So if sickle cell trait in Africa is a protective mechanism against malaria, how does that play into the extraordinary difference that you showed in the distribution of falciparum versus vivax in Africa?
Vivax is not frequent in many areas of sub-Saharan Africa, although recent studies have shown that there are levels of Vivax that haven't been appreciated before. The resistance to Vivax infection in Africa is associated with the lack of a Duffy receptor antigen on the surface of the red cell, and that Duffy receptor antigen is important for the invasion of the Vivax into parasites. It has nothing to do with sickle cell. It has everything to do with the Duffy expression of the Duffy antigen on the cells, and that's well established through studies. Although there are always exceptions to rules, and one of them seems to be that Vivax in areas in East Africa or out near Madagascar and the like, there are strains that seem to be able to invade the Duffy negative red cells. And more work needs to be done. Although, credible resistance to invasion does not have to be absolute for a barrier to effective transmission.
Are there any other questions? Okay, well, listen, I want to thank you both for, whoops.
One in the back.
Do you want to ask a question? Come on, you can ask the question. Speak up.
Thanks, you have great work. We are trying to put together a liver chip to look at some gene expression systems, and I have not seen anything that works well with non-parenchymal cells and liver sinusoidal endothelial cells and gene expression. I'm wondering how well your chip works, if you've looked at some of those things, how expensive it is to set up. It's not an easy question or questions that I'm asking, which was why I was happy to talk to you at the end.
Yeah, I'm happy to talk after. We have the 2D chip I showed you and a perfused chip and the 3D chip, and they all have been characterized, kind of at different levels for different applications.
Great.
Yeah.
Thank you.
Okay. All right. Well, thank you very, very much for a very exciting presentation.